right, well, thank you very much. My name is Annette Kim. I'm at the Brigham and Women's Hospital. But I'm here not to talk about that stuff, but I'm here to talk to you as a member of the CAP Molecular Oncology Committee and some of the work that we've been doing with proficiency testing. So I am a current member of the MOONC committee, and all the data I'm going to present is from that committee. However, they are completely not responsible for any of the snarky comments and attitude that comes out of my mouth. That's all me. It's on me. I do receive some consulting dollars, but that doesn't affect any of the content of the talk. All right, so this is the crazy long outline with all the 50 billion acronyms, and I've even worded some of them as like this against this, and it's really meant to be highly competitive, but this is the United States, so everybody still gets a trophy, so it's okay. Most of this work has been published previously, with the exception of the last little bit there, and so I may refer to you for the sake of time to some of the actual papers. But the point of this, and so let me back up for a second. I will say that at this conference, it's amazing to hear of like all the really cool, sexy stuff that's going on with copy number and structural variants, and you're going to get none of that in this talk. These are going over completely pedestrian assays like BRAF assay and EGFR assays, but I think that the lessons learned here are really important for anyone doing molecular and cytogenetics in this modern era, and I think that the sort of the issues it brings up are relevant throughout for all of us. All right, so in 1906, the Pure Food and Drug Act was passed by Congress, and the FDA was formally created in 1930. 1938, the Federal Food, Drug, and Cosmetic Act extended the umbrella of the FDA to include medical devices. And then in 1976, the Medical Devices Amendment spread that umbrella to include in vitro diagnostics, or IVDs. So they're counting in vitro diagnostics as devices. And I think it's really important to notice that they spread this umbrella to include testing diagnostics well before testing is what it is today, because PCR wasn't even invented until 1983 by Kary Mullis, may he rest in peace. And so I think that's very important, that they could not have possibly envisioned what molecular would look like today when they passed that act. All right, in 1988, the Clinical Laboratory Improvement Act was passed by Congress, and this gave CMS the ability to regulate the accreditation, inspection, and certification of laboratory processes. And even though this is different than regulating the devices themselves, the FDA allowed enforcement discretion and it allowed CLIA to take over some of the oversight of processes of labs. But again, I think it is really important to note that almost 40 years earlier, pathologists were already regulating themselves. We are the ones who care about the results that we're putting out on our patients. We are the ones who are concerned about giving best patient care. And so over almost half a century earlier, CAP started producing proficiency testing. And this is not to tout CAP's horn. This is simply to say that pathologists are the ones who first cared about it well before Congress even knew this was an issue. All right, 2000 was a landmark year which hailed the first draft of the human genome, as well as the launching of the first commercial NGS platform by MPSS, which later became Illumina. So again, the genomic era comes into being, and then all of a sudden the FDA says, oh, wait, now we're going to regulate LDTs. You know, these are all in vitro diagnostics, and so these laboratory developed tests have to come under us, and we're not going to do this enforcement discretion anymore. And in late 2017, the first NGS assay was officially FDA approved, and in early 18, CMS said, okay, we are going to reimburse for that assay, which sets the unfortunate precedent, which is really scary, that they will only reimburse for FDA-approved assays. This lays the burden on us. So what is an in vitro diagnostic? Well, the FDA describes it as a device, which they mean instrument, apparatus, implement, machine, contrivance, implant, or in vitro reagent. So this is, by sticking in those snarky little words right there, those little words, they hooked us. They describe a laboratory-developed test as an IVD that is intended for clinical use and designed, manufactured, and used within a single laboratory. And I have a whole set of snarky comments about that as well, but I think for the sake of time, we'll move on. All right, so the FDA has published all these things that they perceive as being gaps in the CLIA oversight of LDTs. And because I'm backwards, I'm just going to start from the bottom up and address them all. So while it is true that CLIA doesn't necessarily address all of these things, there are things in place that regulate all of these aspects. So they say CLIA does not require informed consent for patients who participate in LDT clinical studies. Yeah, but like we've got the IRB, right? That's what's supposed to regulate that. Assessment of quality of manufacturing of devices, and if you think devices are assays, well, that's part of routine QA/QC in all the laboratories. We've got positive and negative controls on all of our runs. We're constantly assessing this. Requires removal of unsafe devices from the market. Well, CAP has a requirement that extends for almost all of its surveys that if the laboratories fail 2 out of 3 proficiency surveys, they have to cease and desist testing until this problem is rectified. So it's not as if this isn't being regulated by somebody. Requires adverse reporting. Yeah, CLIA doesn't require that, but our hospitals sure do, and so do our QI/QM programs in the laboratories. So this is being regulated. And then lastly, they claim that CLIA does not assure the safety and effectiveness of LDTs, and they talk about the accuracy of testing and analytical and clinical validation. Well, for the vast majority of tests that are produced in most clinical laboratories, we are testing for analytes that have proven clinical utility and validity already. We're not like out there coming up with a signature of high blood pressure or something like that. We're just doing BRAF mutations, right? And so that's really not an issue. And then we use proficiency testing all the time to both help validate our assays and then once we go into production with them to do ongoing quality assurance. And so let's talk a little bit about proficiency testing. Proficiency testing can be done in many different ways, blinded samples, sample swaps, but one very common utilization is for external quality assurance. And CAP is a huge vendor of external quality assurance samples where they send an unknown sample out to all the laboratories, everyone sends in their results, and then we collate those results and say how well labs are doing. And labs that aren't doing well, if they don't do well 2 out of 3 times, they get penalized. So it's an indicator of quality with the goal of course, optimal patient care. So can we use existing PT data to address the perceived issues in analytical and clinical accuracy? So we wanted to look at this FDA LDT issue, and so we turned to the assays that had already FDA-approved companion diagnostics. And these include BRAF, EGFR, and KRAS during the time frame of the study. And you can see here that the FDA-approved companion diagnostics and the drugs for which they are designed. So because of the FDA approval times, the BRAF study starts in 2011 and goes to 2015. And for EGFR and KRAS, it's 2013 to 2015, all right? So slightly different sets of data, but it basically is started from when there was an FDA-approved assay. So let's compare. Let's start on the far right here with KRAS. We had over 2,000 responses on KRAS results, and there was over 97% accuracy. In other words, if it was wild type, they called wild type. If there was a particular mutation, they correctly identified the particular mutation. If we break it down now by LDTs in green and FDA companion diagnostics in yellow, and this color scheme persists throughout the talk, you can see that there's no statistical significance between these. I've really expanded up the top range. The y-axis here is 80 to 100%. For EGFR, there was a statistically significance between the two, with FDA companion diagnostics coming out on top, marginally better at identifying EGFR mutations. But on the flip side, LDTs come out on top for BRAF. So it's a bit of a wash between the two, but it's highly statistically significant here. So we're going to go through in detail what the mutations actually were. So in the case of BRAF, both LDTs and FDA-CDs were equally good at identifying wild-type sequence and the V600E single nucleotide variant. But look at what happens when you go to V600K, the dinucleotide variant. And we can go, LDTs win. All right, so they get a trophy there. So is it just that the FDA assay is terrible? Well, of course not. It wouldn't be FDA approved if it was terrible. It has to do with the design of it. So this particular FDA companion diagnostic has FRET probes that are complementary to either wild-type sequence or to the V600E single nucleotide variant. The FRET probes are attached with different floors, and so as polymerase comes around, the exonuclease activity clips it off, and you get a different color, depending on whether you're wild-type or V600E. And it's actually really great at detecting V600E. And there's a threshold of about 10% neoplastic cellularity, which means it's getting down to about 5% variant allele fraction. Great. However, the V600K is a dinucleotide variant. The FRET probe doesn't bind as well, and so the neoplastic cellularity, the minimum neoplastic cellularity has to be 60% for it to bind at all. And when it does bind, it misidentifies it as a V600E because it produces a blue color, all right? So this is a fault in the design of the assay that we knew about, and so on the MOONC committee, we created on the result form a does not discriminate option. So if your assay does not discriminate between different variants at the same codon, you're supposed to click that. But 35% of labs apparently can't manage to do that. And so this is on the labs. So in some cases it might have been a low variant allele fraction where it wasn't detected at all, and then they get it wrong because they say it's negative and it's positive. Or if they did not say does not discriminate and they misidentified it as a V600E, they get it wrong. And so this comes to the fact that these laboratories probably don't know their assay well enough to realize that it doesn't discriminate, and that's on the labs. But I think the design is on the vendor, right? Because let me ask you this, before you test a specimen, do you actually know what the mutation is? Of course not. Then you wouldn't need to test it, right? So there's no way that you know ahead of time that you have a V600E, so how do you know whether or not your assay is applicable to it ahead of time? There's no way to know. So that sort of limits the clinical utility of this particular assay. All right, EGFR is where the FDA companion diagnostics were slightly better at detecting the variants, and that was completely driven by the L861Q variant, where several LDTs were unable to identify it correctly. We looked into the methodologies used by those LDT laboratories. We looked into the sequence of L861. There's no sort of red flag as to what could be going on there. And in fact, in subsequent surveys that were outside of our 2013 to 2015 study period, there was absolutely no difference between FDAs and LDTs. The LDTs did just fine identifying. So it seemed to be a fluke in one survey. But in all fairness, because we're going to be fair and balanced here, we're going to give the FDA a little trophy here. All right. KRAS, there was no statistical significance at all between LDTs and FDAs. So as you know, with CAP, we don't actually just ask for a result. We ask for various of your practice parameters as well, because it's our job as pathologists to make sure that we are all promoting best practices in everything that we do for patient care. And so we asked, what kind of tissue preparations are accepted, and you can see that there's a large number of laboratories that accept a multitude of different tissue preps, whether it be snap frozen, fresh tissues, FNAs, et cetera. And this is despite the fact that the FDA companion diagnostics are only validated for FFPE, only FDA approved for FFPE. Similarly, when we look at tumor types, we see that more than half of the labs are all accepting a wide range of different tumor types, even though the FDA companion diagnostics are only approved for a single neoplasm. We also looked at neoplastic cellularity. So here's percentage of laboratories responding, and then on the wow, the floor is all vibrating, everything. Along the x-axis, you see the percentage, the neoplastic cellularity, the minimum neoplastic cellularity that is required to run your assay. The red bars here are the minimum tumor burden required by the FDA-approved companion diagnostics. And what you can see is that at the low end, there are several FDA-approved laboratories that are accepting much, much lower neoplastic cellularity. And then finally, other practices were examined as well. Pathologists reviewed DNA quantification, tissue dissection, and I think there was no specific trend for FDA labs or LDT labs to have overall better practice. Oops, sorry. But you can see that these numbers are not 100%. Right? And so all of those labs that are not following the procedure that is FDA approved are doing it off-label. This includes things like the method of DNA quantification, where one of the BRAF assays requires that you do use nanodrop. All right, so not everyone was using nanodrop. Another EGFR required that everyone use spectrophotometry, and not everyone was using spectrophotometry. So, as you know, FDA approved assays have a completely locked down procedure, which means any modification from that lockdown procedure is essentially off-label use. It is an LDT, and it needs to be completely validated as an LDT. So when we tallied up all of those deviations from the FDA-approved procedure from all the different labs, we found that more than 60% of labs using an FDA companion diagnostic were actually using it as an LDT. So to summarize this section, both LDTs and FDAs exceeded 97% accuracy when combined across all 3 analytes. So they all did really, really well. The most significant difference was due to very methodologic specific differences in the detection of BRAF. Greater than 60% of FDA labs are actually using them as LDTs. And I think that this is a really huge point here, which is that real-life clinical practice doesn't follow an FDA lockdown procedure. So your clinicians are going to call you up and say, I can't get a big Goomba of this tumor because it's in a very unfortunate location, so I can't get you an FFPE block. Would you please accept this FNA for testing? Of course we want to accept the FNA for testing, right? Or they may say, you know, I really want to use vemurafenib in this colon cancer. Can you please test that for BRAF? Of course you want to test it for BRAF. Or you may not have money in your budget to buy a nanodrop and you want to use your 30-year-old spectrophotometer. These are real-life exigencies in the lab that are just not taken into account by these lockdown procedures. And so I think it's really important, finally, to know your assay and know some of the assay-specific performance parameters. So one of the most horrifying aspects of this entire procedure was we went to publish this in JAMA Oncology, and JAMA Oncology asked me to produce a tweet. Now, I am an old fogey, and I plan to be the last human being on Earth not on Facebook, and so I was totally mortified at having to tweet out my paper. But here is the tweet I came up with. LDT and FDA-approved assays, overall equivalent performance. Most labs with using FDA assays are using them off-label as LDTs. That's what I said. This is what I really wanted to say. LDTs, you're the best. LDTs forever. Please note that my son had to misspell all of these words for me. So one thing that's really cool about the tweeting thing, I will say that this is cool, is that the journal then tracks all the tweets. And so you can go back and see what people are tweeting about your paper. And so what was awesome is that Dr. Shulsky, who's very prominent in ASCO, said, very reassuring, at least for these tests. Need same rigor for NGS testing. So you ask and we will provide, right? So how are we able to tell which were the FDA labs and which were the LDTs? Because we asked for the manufacturer information. So we can infer some information about who might be using NGS during this time period. And they appeared to do really well. This is a very complicated slide, and I apologize, but the FDA's labs are over here, the individual manufacturers are over here and all combined. And then LDTs are over here and all combined right here. And again, the scale's 80 to 100, and you can see that almost all manufactured kits here are doing, or no kit specified, are doing really well at over like 95% accuracy. And we can even we can infer that someone who's using Illumina or ion torrent reagents are probably doing this by next generation sequencing. And these guys all did extremely well with just a single divot here at KRAS, and that is the result of one lab missing one KRAS variant. So it's really not a hugely significant problem. It seemed to be doing well, but we were inferring who was using NGS. And in fact, there's a whole bunch of labs that say no kit specified. So if you bought your primers from IDT, you might not be represented here. So what we did in 2014 is we introduced a question into our surveys saying, are you producing these results by NGS? And that allowed us to very discreetly discriminate the NGS labs from the non-NGS labs. So we went back through the same process again, looking at BRAF, EGFR, and KRAS. And you can see that NGS assays outperformed non-NGS assays, and that was largely driven by the V600K differential shown here. V600R is also another dinucleotide variant, and there's a suggestion here again that NGS did better. And so NGS wins, yay. All right, EGFR. NGS outperformed non-NGS for the detection of wild type, although they're all like super high. But then also at the L861Q locus, but this also encompasses that one survey where, for whatever reason, many LDT labs had a hard time with this variant. But NGS wins here again. Again, everyone does like phenomenal on KRAS, so we don't need to talk about it. And then we get to the practice component. So it turns out that NGS performing laboratories tend to have overall better laboratory practices in terms of pathologist review of tissue ahead of time, tissue dissection, providing interpretive comments. There were no significant differences in neoplastic cellularity, minimum neoplastic cellularity. But overall, for these other parameters, NGS wins. And so I think that led us to want to bring on, of course, NGS proficiency testing, because now there are a lot of people using it. It's a very good test. And so we actually, in 2014, when we started asking them, are you using NGS? We also asked them, well, so how are you using NGS? And so we asked a bunch of practice questions, which were summarized in a paper, and that allowed, using those answers, that allowed us to design 2 surveys, the NGS Solid Tumor Survey and the NGS Heme Malignancy Survey. So the results of the original questionnaire, the practice questionnaire, are summarized in this paper, and I'm just going to go over a couple of high-level points because many of these may have changed since 2014. At that time, 95% of labs were doing tumor-only sequencing. 60% of laboratories were doing their sequencing on an ion instrument, compared to 40 for Illumina, so we get, ion gets to win there. Methodology, almost everyone was using amplicon-based chemistry. I think this is likely very different now. Variant types, everyone was picking up SNVs and small indels, but only roughly 15% were doing CNVs and structural variants. And again, we've seen so many talks during this conference about the ways that people are now using NGS to do all of these things. And the limit of detection was about 5 to 10% VAF. So when the first results came out in 2016, we were all literally like pouncing on the results, just so eager to see how labs are doing. We were so eager that we immediately published the solid tumor survey, just based on one survey. So that first survey had 10 variants shown here. And so here's the C dot and P dot. Here are the number of labs that test for that particular variant and how they did. And then we'll talk a little bit about the VAF and coverage on other slides. But the overall accuracy was 98.3%, so this is sensitivity for a given variant, for the variants. And the VAFs that were assessed were 15 to 50%. So this is a pictorial view of that 98% accuracy. Blue are all the labs on the y-axis and that got the variants correct on the x-axis. Gray means that lab didn't test for that particular variant. But there were 17 false negatives, and those are highlighted here, where including 3 of which would be reporting error noted here in orange, where the variant was not detected, but another variant in the same codon was, so this is likely a post-analytic reporting problem. There was no clear telltale sign of what caused the false negatives in the remaining 14 cases, and there were no false positives. So overall, phenomenally excellent results. Here's the deviation from the engineered digital PCR VAF, and so they all hover right around 0, and in fact, all the VAFs were within 10% of the engineered fraction. So phenomenal accuracy even at the quantitation level by NGS. And the depth of coverage was quite variable, but many of the labs had over 1,000x at these loci. So we were similarly eager to see the results of the heme malignancy, and so we pounced on those results as well, and I was sifting through it. I went, huh. So I waited for the next survey, and I looked at those results really eagerly, and I went, huh. And so eventually, I ended up having to sum up 2 years' worth of data. So the first 4 surveys, 2 surveys per year, and this encompasses 36 variants in 16 genes. JAK2 and KIT had 2 variants apiece. And so we had 2 years' worth of data, 2 replicates for each of the variants that we saw at different variant allele fractions. And I wanted to just make sure that what I was seeing was really what I was seeing. The engineered VAFs were a little bit lower here, so we were assessing almost down to 10% variant allele fraction. And the overall accuracy, if we take the true negative and true positive rate, was over 99.2% with over 25,000 pieces of data. The true negative rate or specificity was 99.8%, and the true positive rate was 93.5% sensitivity. So that is lower than the 98.3 that we saw in the solid tumor assay, and this is largely driven by the issues with the detection of CEBP alpha. So as most of you know, the N-terminus of this is just like an entire string of C's and G's. So it's very difficult to accurately synthesize primers, accurately synthesize probes, and accurately sequence these regions. And so if we just look at the true positives, the false negatives, and the false positives, I'm not going to show you the 22,000 false true negatives. That would make all my pie graph look really, really dinky. You can see what the data looks like. Again, we had 151 false negatives, the largest component, single component of which was CEBP alpha misses. But anytime you have a sample swap, that results in false negative and false positive results. And so a quarter of our false positives were due to sample swaps, which also accounts for a significant number of our false negatives. The other 50% of our false positives were where they clicked the wrong button. They reported the button right next to the correct variant. So we think that this is a post-analytic error. So we have pre-analytic errors, post-analytic errors, and so only 11 out of over 25,000 possible results were possibly analytic errors. So it's really, really low, all right? But what the VAF is going on with the Amplicon platforms? All right, so here's variant allele fraction, and here are all of the variants that we tested for over the course of 2 years. The engineered digital PCR VAF is shown with a red box with an X through it, and you can see that for many of the variants, there's like this weird bimodal distribution of the VAFs, and all the Amplicon stuff is at the bottom. And this was statistically significant in 6 out of the 36 variants. So then we singled out Illumina TruSeq chemistry, and 32 out of 36 variants showed statistically significantly lower VAFs. And so we were like, what the heck's going on with TruSeq chemistry? And so we looked at coverage. Actually, the coverage was actually higher across the board here. There's another option. There's the Illumina TruSight myeloid panel, by which the vendor has chosen the content. And then you also have the option of TruSeq custom panel where you design the content. But across the board, it was the same sort of low VAFs for both options. And so it really is the chemistry, not who designed the content. And so then we did a series of analyses to try to figure out what was going on. We even invented a new mathematical formula of VAF standard deviation index to how far something was off from the mean VAF or the targeted VAF. And what you can see is that it isn't coverage, it isn't what kind of sequencing platform anyone's using, it isn't the read length, it isn't the variant caller that people are using, it's not the CG percentage surrounding the variant. We looked for degradation, so we had people send their samples back, and then the vendor redid the digital PCR, and you can see that the R-squared and the slopes are all 1. There was no degradation. The vendor was very helpful here. Illumina was also very helpful. They offered to reanalyze FASTQ files on the most up-to-date pipeline, thinking that maybe some of the pipelines were throwing out reads that had the variant in it. But in fact, the variants were like dead on for 2 different samples in the reanalysis. But what was interesting is they also looked at the SNPs, and all the SNPs were at 1 or 0.5. They looked like SNPs. There was no underestimation of the VAF here. So this led us to consider what our proficiency survey materials were really made out of. I can't show you a diagram. The vendor has asked that I not show you the diagram, so I have to respect their wishes, but I can tell you about it. All right, so what it is that we've got genomic DNA. All right, that's just normal gDNA that everyone's got, and it's got whatever sequence. Then we have an 1,000 base pair insertion where in the middle of it is the variant in question, but the rest of the backbone there is exactly the genomic DNA sequence. All right, so 1,000 base pairs. Then there's these marker sequences on either side that allow the vendor to manage the insert and put it into a circular plasmid, and then they build that up. And so you've got this circular plasmid here. They linearize it, so now you've got marker, insert, marker, and then linearized plasmid, okay? And that's what they dilute into the genomic DNA. All right, so my thought was that maybe there's some weird secondary structure with all this non-native sequence with the marker and the plasmid and all that kind of stuff. Maybe that's what's going on. So the vendor very kindly worked with us and made us constructs where they were diluted into the diluent DNA was just the insert or the insert plus the marker. And that's shown here as I or IM. And the digital PCR results are in these dark blue for each of the variant, the first 2 bars. What you can see, especially highlighted at the lower VAFs with IDH kit and the other kit mutation, is that those TruSeq chemistry labs got very, very low VAFs for both the insert and the insert plus the marker, but this is especially accentuated when you have the marker, and you can really see that very nicely on NRAS. All right, so that non-native sequence is futzing with the VAF. One of the labs actually had 2 different platforms by which they could test these variants, and so when they did it on their ion torrent, the VAFs were totally fine. So it is something peculiar about some of the non-native sequence and perhaps secondary structure. And if you think about the TruSeq chemistry, when we normally do, it's amplicon chemistry, when we normally do PCR, we've got a 5' primer and a 3' primer on 2 opposite strands, but these have the 5' and the 3' on the same strand, and then there is an elongation and a ligation step. So this is single-stranded PCR, and you might imagine that single-stranded PCR might be exquisitely sensitive to alterations in secondary conformation. And so that's what we think is going on here. But in summary, the overall accuracy of both the NGS solid tumor and heme surveys was excellent, even with quite varied and challenging variants. There are poor performing regions of note, so it's really important to know your assay and to make sure that your clinicians and your clientele understand the strengths and the weaknesses of your assay, whatever it be, whether it be BRAF V600E or CEBP alpha by NGS. It's also really important to know your proficiency materials, because you can get very unexpected interactions between the proficiency material and your unique assay chemistry. And then finally, we're taking NGS and many other of these novel platforms into all new areas. We have some proficiency testing available for some of these already, and some of these we're in active discussions to bring these online. But as we move into things like cfDNA, MRD, copy number variants, structural variants, mutational profiles, I think it is really, really important for all of us to be very vigilant about the materials we use for our positive controls, for our proficiency testing. Be very vigilant about the conclusions we make about the data. And then I think it's absolutely critical that we be out there presenting this data over and over and over again to help inform conversations about regulation and reimbursement. Because we don't want to be in a situation where people just assume that LDTs are all terrible or that NGS can't possibly be any good when the data proves that it is. So finally, I'd just like to thank first the College of American Pathologists in general for selecting me and bringing me onto the committee. The MOONC committee has been just a fabulous group of individuals, and our collaborating vendors were extremely helpful in all aspects of this investigation. The people that are shown on this slide are the people who are actually authors on those 6 papers, and I'd like to give a special shout out to Alyssa Keegan and Leah Surrey, who are junior members on the papers that I led. Tom Long is a biostatistician, and Patty Vazelos is the staffer extraordinaire at CAP, who is just incredibly skilled at herding cats. Jason Merker was the liaison to the NGS working group at CAP, and he really shepherded the onboarding of all of the NGS surveys. And then Joel Moncur is listed twice, not just because it evens out that bottom row, which I think is very important symmetrically, but also because he was my comrade in arms in the LDT FDA papers, and we kind of traded off being senior author on each of the papers. So anyway, and I thank you all for your time.